For this crime scene project, we had to start it by first analyzing an actual crime scene scenario while in class. We had to stand out of the crime scene and call out what we noticed that might be important to the case, by 1 being the first thing we see and 22 being the last thing we saw. Then we had to sketch out the crime scene and label what is where and how far it was. For the polygraph project, we have to download a lie detecting test app to connect to a Bluetooth device that tracks a person's heart rate. Then we pair up with a partner and ask them yes or no questions. First starting with regular everyday questions to find the usual heart rate. Then we ask them 10 yes or no questions we want and write down the responses and what was the heart rate. For the timeline project we did for Anna, we had to go to PLTW and track down everything she had done for the last week before her death. For the project, we wrote down everything that she did or what was listed on social media sites that she was a part of. We had to list any relevant information from text messages or social media in the video that she had posted, and we had to list any important information or detail that was on the video.
In the strawberry DNA lab, we used a simple solution of ethanol and dish soap to isolate the DNA of strawberries and display its protein structures. This helped us learn about the structure of DNA. In the heart dissection lab, we guided blue and red string through the valves of a sheep's heart in order to better understand heart anatomy and how the heart pumps blood. By collaborating with our teams to create a custom board game, we were able to study the anatomy, function, and surgeries of the heart in an easy-to-understand and interactive way.
The calorimetry project was a project where we chose one of our favorite foods and burned it in the calorimeter. This was to find that food's estimated calorie count. More importantly, the point of this project was to learn about the process of finding a food's exact calorie count. This project also opened our eyes to the foods we are putting into our body and what a healthy diet looks like. CRISPR is another thing we learned about in biomedical science. CRISPR, or CRISPR-Cas9, is an interesting technology that allows doctors to alter or edit parts of DNA. This family of DNA was originally found in bacteria. We learned about CRISPR because by the time we are adults, it is likely to be used instead of common practices such as plastic surgery. It is important to learn about the future before it arrives. The Nutrition Label Project was a project where we chose the nutrition label from a food around the house or online to research about it. This project involved learning about what a nutrition label was and how to read one. The purpose of this project was to teach us the many different things on a nutrition label and what we should be looking out for when reading them.